So we have actually looked at the different methods that are being used in gene technology so far, right? In cutting and joining of DNA, inserting to the whole cell, and then culturing the whole cell so that the gene is being cloned and also being expressed. So we also learned about using the gene probe in genetic fingerprinting, using the gene probe in microarray. So these are all the techniques that we have learned so far. So now we want to look at when we apply those techniques in medicine, how does it help us? So the learning outcome says to explain the advantages of using recombinant human proteins. So for example, like insulins have been produced by genetic engineering. So they are called the recombinant insulin. So these recombinant human proteins, we use them to treat disease. For example, insulin to treat diabetes, factor VIII, which is to treat hemophilia because it's a blood clotting factor, adenosine deaminase, ADA, is an enzyme that is used to treat severe combined immunodeficiency. That means the person's immune system is weakened. So we use this protein that's produced, inject to the person's body to treat this disease. So this is a recap of the 6 basic steps in genetic engineering, 6 basic steps to produce the recombinant DNA. So we have step number 1 is called isolation. That means we obtain a copy of the wanted gene from the whole cell. And amplification means after you obtain the gene, we amplify it, means we make multiple copies of it by PCR. Then insert the gene into a vector, which can be plasmid or virus or liposome. So far, you only learn plasmid up to now, to deliver the gene to the host cell. So then we will try to transfer the recombinant DNA to the whole cell. So it's called transformation. Because only 1% of the bacteria will take up the plasmid, so we have to do a selection to select whole cell that have taken up the gene. That means we say to select the transformed bacteria, or if you use other cell, it could be other organism, could be transgenic organism or GMO, genetically modified organism or plant. Okay. Then after that, clone and express the gene. So we want the gene that's inserted to the bacteria to multiply and to be expressed. So we have to culture the bacteria so that the bacteria will divide. So as they divide, they will replicate the plasmid and hence replicate the gene, and then they express the gene to form the product, which we call here as the recombinant protein. So if you can please label here, this one is actually the recombinant protein produced in this way. So please label this into your notes. So what are the uses so far? 3 examples you're going to learn is insulin, factor VIII, adenosine deaminase. All these are that are produced by genetic technology to treat diabetes. So the whole cell that they used for insulin production last time is actually E. coli. It's a type of bacteria, E. coli. Factor VIII, they culture hamster kidney and ovary cells. That means they retrieve or they isolate those kidney cell and ovary cell from hamster, and then they culture those cells in a container in the lab in nutrient medium, just like how we culture the bacteria. So they culture, instead of bacteria, they culture this mammalian cell, which is a eukaryotic cell. Okay, and then for production of ADA, they have used E. coli. They have also used insect larva cell. That means they also isolate those cells and culture in the lab, just like you culture the bacteria. Okay? And then to produce those recombinant protein. So what is the advantage of using the recombinant protein is we can use bacteria, yeast, as well as mammalian cells as the whole cells to produce that human protein for us. So instead of slaughtering the animal and then try to isolate that small amount of insulin or factor VIII and other things from the animal, from the slaughtered animal, it's very little. So this whole cell only needs simple nutrition. So when we culture we culture them in the lab. We just make sure that we provide optimum condition for them to grow, like optimum temperature, optimum pH, supply of oxygen or gas, and then the nutrients where it's in a powder form, we add the water, and then we heat it up so that it will dissolve, and so that we will sterilize this solution so that no other cells are inside. So when you add the bacterial cell will use the nutrient to grow. So this is called the nutrient medium. Okay, nutrient medium. So simple nutrient medium, so easy to grow them, and we can grow them in large volumes. Large volumes can be produced. That means, for example, you grow bacteria. If your normal conical flask, we have 1 liter or 500 ml conical flask. You got 1 liter, you got 5 liters, and you can have 10 liters. So you can actually change the size of the container for you to grow like how much you want to grow, as long as you can control optimum condition in that container, which is called the fermenter. Whenever you use a container to grow any cells, we call them the fermenter. Okay? Crystal, you okay? Okay. So, and so when you grow them in the fermenter, the space that you need is very much less compared to if you need to grow the animals to obtain the same amount of insulin. Okay. And few practical and ethical problems, because the protein is not extracted from animal source. For vegetarian, we don't like that. Okay. And then it's not obtained from donor's blood, whereby they may contain, for example, you want a factor VIII from this donor, and this donor just got a HIV infection. So the blood will contain HIV. So the HIV will be transferred together. So we don't want pathogens to be transferred together with the protein. So that reduces risk of infection. Now, what is the disadvantage? Is we already know, this is a recap, for prokaryotes cannot carry out RNA splicing. So if you just give them the original gene from human, which contain exon and intron, then prokaryote cannot carry out RNA splicing. So cannot remove the intron part. Okay, and then they also have no Golgi body, so they cannot modify the protein. It means join the 2 chains together, let the chain bend and fold into tertiary structure. So all these things cannot be done in prokaryotes. So therefore, to solve that problem, latest methods is they use more of eukaryotic cell like yeast cell and mammalian cells. Even the insect larva cell, okay, it's eukaryotic cell. So therefore, for this kind of cell, they have to use eukaryotic promoter sequence to be put in the upstream of the gene. So these cells got Golgi body, so they can process the insulin to correct tertiary conformation. So this is hemophilia. People with hemophilia are lack of blood clotting factor VIII. So whenever they don't have blood clotting factor VIII, you can see that the thrombin cannot be converted to thrombin to form the fiber-like structure. Okay. Thrombin is a fiber-like structure that will trap the red blood cells and then causes the blood to clot. So the blood cannot clot because lack of factor VIII. So human gene for factor VIII, the name of the gene is F8 in italic. It has been transferred to the cells from hamster, kidney, and ovary. So these cells are cultured in the lab in animal culture medium. So for bacteria, we use different culture medium than animal cell, because different cells need different nutrients requirement. So after culturing, then we will extract factor VIII protein, purify and administer it to the hemophilia. So means people with hemophilia will then get the factor VIII. Now let's look at what is SCID. SCID stands for severe immunodeficiency. Okay, severe combined immunodeficiency. There could be a few reasons that can cause SCID. One of the reasons you will learn it here. So when a person got SCID, what happens is that there is no T lymphocytes in the immune system. No T lymphocytes. For example, T helper cell is the manager, right, of the immune system. T helper cell. So it's the manager of immune system. So when you don't have T lymphocytes, we don't have the T helper cell, then that means to say that the clonal expansion of B cells, clonal expansion of T killer cells cannot happen because they need T helper cell to secrete cytokine or lymphokine. So the entire immune system cannot function. So this person will have got risk of infection and die from infection. Okay. So one of the cause of SCID is adenosine deaminase deficiency. So this kind of SCID is called ADA SCID because it's lack of this particular enzyme. Okay, so what does enzyme do? This enzyme catalyzes the deamination of two components in the cell, which is adenosine and deoxyadenosine. So it will catalyze deamination, means removal of amine, NH2, from these two components. So, and this is during metabolism of DNA because DNA got adenosine, right? So this is part of metabolism of DNA. Now, between these two components, right, deoxyadenosine is very toxic to the cell. So therefore, the process of deamination is to convert it into a product that's not so toxic. So that's why this enzyme is very important. This enzyme. Okay, so therefore, when this enzyme is not working in this person, then deoxyadenosine will not be converted to a harmless product. It remains there, is toxic, so it can kill the cell. So it kills the T lymphocyte. That's why the body is lack of T lymphocyte. And therefore, there's immunodeficiency in the person's body. So this ADA gene is found in chromosome number 20. Because of mutation, it leads to change in base sequence of the gene, change in base sequence of RNA, and therefore change in amino acid sequence of this enzyme. So this enzyme becomes of different shape. So when change shape, substrate cannot fit into active site to form enzyme substrate complex, so no product form. Okay, so you can see that this is linked to your AS. So the question that come out in exam can also ask you something related to AS, like what I've mentioned just now. Okay. So for these people, right, they have to constantly take antibiotics because the immune system is not working. So need to take antibiotics to kill the bacteria and fungi that enter their body. Or another way is they can receive adenosine deaminase injections straight away into the body fluids, so that now the cell has got this enzyme to be able to cause the deamination or to catalyze the deamination of deoxyadenosine. Okay, so if given a choice, will you choose to take antibiotics or you choose to take adenosine deaminase? Yeah, injection, which one? Antibiotic or adenosine? Adenosine. Or I give you another choice. Why not I say that, you know, I can add a normal ADA gene into your cell so your cell can produce the enzyme. That's called gene therapy. Any gene you got defect, right? I just add a new one which is normal, then your cell function normally, right? So can you see that the advancement in medicine? You know, firstly, you use antibiotic because this is the only way we know. Then after that, you discover, hey, actually, it's adenosine, so I just inject this enzyme. Then later, you find that, hey, now there's advancement. I can actually add certain gene into the cell, and the cell can function normally. So that's called the advancement of the technology as they discover new and new methods. Okay, so we'll learn about gene therapy in subsequent lesson. So you have learned in this short lesson here the advantages of using human recombinant proteins. An example that we look at is using insulin. We call it recombinant insulin for treatment of diabetes, recombinant factor VIII for the treatment of hemophilia, recombinant adenosine deaminase for the treatment of severe combined immunodeficiency. So that's all for this topic.